Hi, Ninja Nerds. In this video today, we are going to be talking about the development of the skeletal system. Before we get started on this video, please continue to support us by hitting that like button, commenting down in the comment section, and please subscribe. Also, down in the description box, we have links to all of our social media platforms. You guys can go check that out. And if you guys can donate, Patreon, we would truly appreciate it. All right, Ninja Nerds, let's get into it. All right, Ninja Nerds, so when we talk about the development of the skeletal system, we're going to have two primary goals throughout this video. First thing is we got to develop the axial skeleton. So what does that consist of? We got to talk about the development of the skull, okay? And we'll talk about what's called the neurocranium and the visceral cranium in a second. After we talk about the development of the skull, then what we're going to do is we're going to talk about kind of the development of the actual thoracic cage, right? Particularly the ribs, the vertebrae, and the sternum. And then we'll finish up talking about the development of the upper limbs and lower limbs, okay? So let's go ahead and start talking about the skull. So the first thing that we have to understand is we're going to take which layer to make the skull. Do you guys remember from the videos that we've been doing here? Remember the mesoderm and the neural crest cells? Those are very important for developing some of the structures of the skull. The first one that we're going to talk about is what's called the neurocranium. The neurocranium is the part of the skull that encases or surrounds the brain. So where does the neurocranium, which we're going to talk about here, so the first one that we're going to discuss is the development of the neurocranium. The neurocranium is going to develop from what structures? Well, here, we got to take our trilaminar disc. From the trilaminar disc, what is it made up of? I know you guys remember this. The top layer is going to be the ectoderm. The middle layer is going to be consisting of the mesoderm. And we have all the different components of that mesoderm, like the different subsets. And we'll talk about those. And then the last one that we said, the most inner layer here, is going to be the endoderm. Now, kind of center, axial, right in the center of the mesoderm, you have a couple structures. You have this purple structure here. What is that called? This guy is called your neural tube. It's called your neural tube. And then you have the notochord, this little like black dot that's going to be anterior to the neural tube or just above the endoderm. Then on the sides here, what is this particular part of the mesoderm called? That's called your paraxial mesoderm. That's very, very important. We'll talk about that in a second. And then on the sides of the neural tube, on the top, what do you have? The neural crest cells. Now, here's what I want you to do. We're only looking at a cross-section of the embryo. What I want us to do now is take this neural tube and just yank it out. Okay, so I'm going to take the neural tube and I'm going to yank it out and I'm going to smack it right there. Then I'm going to take the neural crest cells, which are on the sides of the neural tube, and I'm going to yank those out and put those on the sides. Then I'm going to take these chunks of paraxial mesoderm and yank those out and put those on the sides. Now, let's talk about these things. So first thing we have here is our neural tube. We're just starting to see the vesiculation here. Don't worry about that yet. But what we have on the side of the neural tube is these little brown dots. What are these little brown dots here called on the side of the neural tube? This is called your neural crest cells. These are called your neural crest cells. And these are going to be towards the top of the actual neural tube. Then these red structures here, which we said was particularly what? The paraxial mesoderm. This is called your paraxial mesoderm. Remember we talked about in the mesoderm video, the paraxial mesoderm undergoes this kind of segmentation process where it forms somites. Well, the somites that are up towards the top, right, are actually what type of somites? Occipital somites. So the particular part of the paraxial mesoderm that we really need to know is called the occipital somites. What are those again? You have a chunk of mesoderm, paraxial. They start segmenting, making somites. The ones towards the top are occipital somites. If we take the neural crest cells and the occipital somites and combine them together, guess what we get? The neurocranium. So what these things start doing is these cells start migrating around the skull and they start making a bony covering around the actual brain. And what is that called? Your neurocranium. Now we got to talk about the parts of the neurocranium. So you have the base of the neurocranium. There's a very, very important name for this. The base of the neurocranium. So what we're doing is I'm chopping the skull like this. I'm chopping it, looking into the skull. And what I see is, I see a couple different fossas, if you will. This right here is called your posterior fossa. This one here is called your middle cranial fossa. So you got posterior cranial fossa, middle cranial fossa, anterior cranial fossa. There's a bunch of bones that make up this base of the skull. What are those bones? You have the occipital bone. What else do you got? You see this kind of bony part here that goes like this? This is called your petrous part of the temporal bone. What is this called? This is called the petrous part of the temporal bone. All right, so we got two bones so far. We got the occipital bone, and then we got the petrous part of the temporal bone. Then you have this bone right here. All of these bones that are kind of coming off of this point here and moving outwards. This kind of gives like a bat-like appearance. It will kind of extend from here to here, all this in between. This is called the sphenoid bone. So this is called your sphenoid bone. And then last but not least, you have this bone here right in the front, this little kind of like thing coming up called the crista galli. But this whole bone here is called your ethmoid bone. These bones are what make up the base of the skull and are a part of the neurocranium. Here's what's very interesting though. This is what's very important. This neurocranium is also given another type of subname, if you will. And the subname for it is called the Chondrocranium. The reason why I want you to remember that is chondro means cartilage. So chondrocytes. What we have is the paraxial mesoderm, and we have those neural crest cells. Technically, these become what's called multipotent mesenchymal cells. And these mesenchymal cells undergo a specific type of process where they differentiate. And when they differentiate, they turn into a special type of cell called a chondrocyte. Okay, so we got our mesenchymal cells that turn into a chondrocyte. Then what happens is this chondrocytes create the framework and then start actually differentiating into what? Bone. So then your chondrocytes start converting into what's called bone, particularly what's called osteoblasts. This process where you go from mesenchymal cells to chondrocytes to osteoblasts is called what? Endochondral ossification. So this is called endochondral ossification. Endochondral ossification is important because most of the bones are developed. Most of our skeletal system is developed by this process. Very important to remember, especially the chondrocranium component of the neurocranium. The other type is this here. We've only talked about the base of the skull. Remember I told you I took a section like this and I looked into the actual skull cavity? Well, that part that I cut right here, the part that I'm removing, the calvaria, that right there is going to be this part that we're staring at. This component here is also a part of the neurocranium. But again, we got to give it a special little name. So we call this one the chondrocranium, the base of the skull. The top of the skull is actually going to be called something very interesting. This one is called the membranous neurocranium. The membranous neurocranium. This is the chondrocranium, or the chondroneurocranium, and this is the membranous neurocranium. So what kind of process do you think these actual cells here, what kind of cells? The paraxial mesoderm, which part though? The occipital somites, and the neural crest cells. They go a differentiation process. But this time, guess what they do? They convert straight from the mesenchymal cells to bone tissue, osteoblasts, and then start making bone. This process here is called intramembranous ossification. Intramembranous ossification. And not many bones in the body are developed by this process. One of the few is the top of the skull. Now, what bones make up this? And then one last thing that we got to talk about. The bones that are developed by this process of intramembranous ossification is this bone right here in the front. You know you have a suture here, and these are very important structures during the baby whenever they're developing. We'll talk about those in a second. But here, this bone in front of this coronal suture here is called your frontal bone. So that's developed by the process of intramembranous ossification. Then the bones here on the side of the sagittal suture. This is called your parietal bone. Okay, you have two of these bad boys though, right? And then this one here, in between this lambdoid suture, is called your occipital bone. Now you guys might be like, wait, Zach, you said the occipital bone was developed from the endochondral ossification up here. That's true. But if you take, again, that cut here, where you're kind of slicing through, you're going to have a piece of that actual occipital bone that you're going to take from the calvaria. But again, you're also going to have the base of the occipital bone underneath, where the occipital condyles come off as well. So you're going to get occipital bone, the top of it developed by intramembranous, and the base of it developed by endochondral ossification. Okay? Now, again, I want you to remember these sutures because they help to separate things. So this suture here is called your coronal suture. Okay, this is called your coronal suture. These are important clinically, that we're not going to go into all of it, but there is importances to these actual sutures if they don't actually completely, if they close too early. We call them craniocystosis types of conditions. But this suture here, which separates the parietal bones, is called your sagittal suture. And then last but not least, this suture here is called your lambdoid suture. Okay, and these respectively kind of separate all these bones from one another. One more thing is these little puffy things, these little open areas of the skull that are very, very important when the baby is developing. These little areas where more than two bones kind of come together, so you have the parietal bones and the frontal bone kind of coming together, you make this little kind of opening here, which kind of allows for the baby's skull to continue to, like as the brain continues to grow during embryological development, you have to have the skull kind of grow to accommodate that. Also, when the baby's being born, that skull needs to be able to kind of move and glide a little bit. And so sometimes these fontanelles allow that to happen. So here we have this fontanelle, which is where more than two bones are coming together, in this case frontal and the parietals. This is called your anterior fontanelle. And these can also be monitors of intracranial pressure. If there's high intracranial pressure, what do you think that fontanelle is going to do? It's going to bulge out. And if there's low intracranial pressure, what do you think the fontanelle is going to do? Bulge in, right? Then you have the fontanelle back here. This one here, we're going to swoop it around here in a second, but this right here is where the occipital and the parietal bones meet. What is this one? This is called your posterior fontanelle. Now these eventually close. This one closes first, and this one closes a little bit later, okay? But again, knowing their importance is for allowing for the brain to grow, we need to allow for the skull to continue to expand, and also kind of that flexibility during the birthing process. All right, beautiful. We talked about the neurocranium. Let's hit that visceral cranium. All right, engineers, so we just finished talking about the development of the base of the skull, right? The chondrocranium. And then we talked about the top of the skull, the cranial vault, which was the membranous neurocranium, right? Now what I want us to do is talk about the structures that are developing for the face, which is your visceral cranium, okay? So now let's talk about the good old visceral cranium. Now the visceral cranium develops a little bit differently than what we talked about. You would think, oh, it's going to develop from the mesoderm. Well, it actually develops from some structures called the pharyngeal arches. We'll go over these in more detail in the future, but what I want you to think about here is we have a sagittal section of the embryo, right? And you can still see the ectoderm, you can still see the mesoderm, and then you can see the endoderm. But you're seeing this in kind of like a different way. A sagittal section here is showing you kind of from cranial to caudal fashion. Okay, whereas the cross section, you're kind of seeing that in a different view. So what I want us to do is take a section about right here, in the top of this tube here. And what you're going to do is you're going to kind of cut this and zoom in on it. And what you get is this very specialized structure. We'll go into this in more detail, but this is going to be called your pharyngeal apparatus. And you see these like little kind of like vesiculations here, how you have like this vesiculation here and then this one here. This right here are called your pharyngeal arches. Okay, so you have these pharyngeal arches. The one that we care about for the development of the visceral cranium is your first pharyngeal arch. Okay, so the first pharyngeal arch is going to be the structure that's going to develop into the visceral cranium. There is a teensy bit of the second pharyngeal arch for one little bone, and we'll talk about that one. But other than that, for the most part, all the structures of the visceral cranium, a very teensy bit of the second pharyngeal arch, but mainly the first pharyngeal arch is going to be the structure that develops into your visceral cranium. All right, and the same process happens here. If you look at the pharyngeal apparatus here, I kind of color-coded it. So you can see the outer part of the pharyngeal apparatus, this blue color is ectoderm. The big chunks here in the middle is your mesoderm. And then this inner chunk here, which is going to be what's called the primitive pharynx, is going to be the endoderm. The mesoderm chunk of this pharyngeal arch is going to be what really turns into the visceral cranium. And it's the same process. We'll talk about, again, that mesenchymal to cartilage to bone process in a second. All right, for the visceral cranium, the main structures is the bones of the face. And they develop in kind of like these processes where you have the first pharyngeal arch. It kind of gives what's called a ventral and a dorsal component of it. But what happens here is you're going to get some bones that are going to be made from this. What are those bones? This bone here that's made from the first pharyngeal arch is going to be called the mandible. Okay, so you're going to form what's called the mandible. The other one here is going to be this bad boy up here on the top, and this is going to be called your maxilla. Then you have this bone up here, your cheekbone. What is that bone here called? This bone here is called your zygomatic bones. Okay, then you have another couple bones here left. Okay, back here you have this bone portion here. It's kind of coming up like this. There's a little suture here. We can't really completely see it, but it's called your squamous suture. Okay, below that squamous suture is going to be a part of the temporal bone. So what is this part of the temporal bone called? This is called the squamous part of the temporal bone. And on top of that, there's also another little portion here. You see this little hole here that's coming on that squamous part of the temporal bone? This structure right here is actually called the external acoustic meatus. These are all developed from the first pharyngeal arch. Now again, going back to this, what is the way that they're going through this process? Very, very important. All of this is happening through a very specific process. You're taking these mesodermal cells. They're differentiating. What are they differentiating into? Straight into bone. So if they're going from the mesodermal cells straight to bone tissue, let's go ahead and recap you guys' brains on this. What is this called? This is called intramembranous ossification. Beautiful. Now, intramembranous ossification, we already said what parts develop from the top of the skull and then including the facial bones. There's only one other area, the clavicles. Everything else is endochondral ossification. All right, beautiful. There's a couple other components that I just want to mention that are developed from the first pharyngeal arch. Let's come down here. What are they? All right, so you have some bones that come from the actual first pharyngeal arch and move their way down into the neck. Make a couple bones here in the neck, right? What are those bones? And cartilage. The first bone here is going to be what's called the hyoid bone. So one of these bones here is called the hyoid bone. The other that are not bones are these blue components here. These blue components actually are going to be what's called what? The laryngeal cartilages, right? And when we talk about these laryngeal cartilage, so the laryngeal cartilage, what are the two components of the laryngeal cartilage that we actually see here? There's a bunch of them. We're not going to go through all of them, but I'm going to tell you. There is the thyroid cartilage. There's the cricoid cartilage. There's the arytenoid cartilage, right? And then there's the cuneiform cartilage. But all of these are developed from the first pharyngeal arch. So we got the hyoid bone. We got the laryngeal cartilage. Guess what else? Little bones that are in your actual middle ear. What are these? Two of them here are developed from the first pharyngeal arch. What is this one? This one right here looks like a mallet, right? So we call this one malleus. And this one here looks like kind of a little anvil. We call this incus. There's one other one, and that's actually the one that's developed from the second pharyngeal arch. Okay, I'm going to do this one in a different color. So again, I want you to remember that this is not from the first pharyngeal arch. This is actually from the second pharyngeal arch. What is this one here called? This is called stapes. But again, what do I want you to remember? This is from the second arch. Okay, very, very important. All right, for embryological sake. Now, what's important also to mention here is that the mesodermal cells that actually convert into this bone, particularly the bones, go through a very interesting process. You take the mesodermal cells, what do? They undergo a differentiation process. When they differentiate, what do they turn into? They turn into cartilage. Then from the cartilage, then what do they turn into? Some of it will stay as cartilage, right, like the laryngeal cartilage. But the other components, the hyoid bone, the malleus, the incus, and the stapes will do what? They'll convert into bone. And when they convert into this bone tissue, what is this process here called? This is called endochondral ossification. Okay? All right, beautiful. So what have we done so far? We made the base of the skull. We made the top of the skull. We made the face, and we made a couple other bones in the neck and in the ear. Now, let's go on to talk about the trunk. All right, Ninja Nerds, so we've talked about the skull. Let's go to the trunk. Let's start making our vertebrae. Let's start making the ribs. Let's start making the sternum. So, from this process, we're going to have to, again, take a look here at our developing embryo. When we take a look here at our developing embryo, what do we have here? Again, you got your neural tube, you got your notochord, you got your paraxial mesoderm here on the sides, you got your intermediate mesoderm, and then you got your lateral plate mesoderms, right? What I really want us to focus on is what part. I want us to take this neural tube out and yank this sucker out, and then take the paraxial mesoderms and put it here on the side, okay? Before we go through this kind of what's called resegmentation process, I want to talk about what happens to this paraxial mesoderm. All right, so now before we start going through this kind of like resegmentation process, what I want us to do is, again, we're going to take the neural tube, and on the sides you got your paraxial mesoderm. I just want you guys to understand what actually happens to this paraxial mesoderm a little bit. So let's go ahead and take and kind of zoom in on pulling out that notochord and the actual paraxial mesoderms on the side in a cross-sectional view. So if we look at this, what do you have here? Again, you got your neural tube here, right? You got your notochord right there in the center. On the sides, you have your paraxial mesoderm. What happens is the paraxial mesoderm starts to develop these like little holes in it. And what is that called? So this is going to be your paraxial mesoderm. And what happens is they start forming these little segments or chunks along the length of the notochord that are called somites. But these somites start developing these little holes in them called somatoceles. So this is a somite. A chunk of paraxial mesoderm is called a somite, but this right here is called a somatocele of the somite. Okay? So you got your somatocele. What happens is this somatocele continues to keep expanding and expanding and expanding. And as it does, it cuts this actual somite into two chunks. One is a dorsal chunk and one is a ventral chunk. This dorsal chunk here is going to be what's called the dermatomyotome. And this ventral chunk here is going to be what's called the sclerotome. Then what happens is the dermatomyotome even cuts in half again and forms into two separate chunks. So now this becomes the dermatome on the back, which is going to obviously make your dermis and subcutaneous tissue. Underneath that, you're going to have muscle. So this is going to be called the myotome. Later, there is another one, we'll talk about that in the muscular system, called the syndotome. Okay? But then this one right here is going to be the sclerotome. Man, this stuff is so cool, man. Isn't it crazy? From ninja to nerd, I want you guys to comment where you guys are at in this video. All right? Make sure you do that. All right, but what I want you to remember here, with the sclerotome is the one that I really want you to focus on here with the development of the axial skeleton. This is the one that actually is going to come around and make the bones, the vertebrae, the ribs, all of those things that are coming off. So the sclerotome, please don't forget that one. So, we took the somites, we went through this process of explaining it. What I need you to next understand is, let's say here we have the somites. Okay, we pulled the somites out. But I want you to remember what part of the somite is actually going to make the vertebrae and the ribs. It is the sclerotome. here we have the somites in chunks. If we kind of cut them in half, let's color code this. The top part is going to be the rostral portion, okay? And then this brown part here is going to be kind of the caudal portion. So you have the caudal portion here, okay? And then here you're going to have the rostral portion in blue. So again, rostral is blue, the caudal is going to be brown, okay? What happens is in this situation, whenever your actual neural tube continues to develop, you have to have the nerves that come off of it and move through the vertebrae, right? And right now, the way this is, there is no intersegmental portion for the actual spinal nerves to move out from the neural tube. So what happens is a very special process that these somites go through, which is called resegmentation. All this is to allow for this vertebral space for the actual nerves to run through. How does that happen? Very interesting. What happens is this actual caudal portion will actually get fused with the rostral portion. So each one of these portions are going to move up and fuse with the one above it. Okay? So now what happens here? Let's look. The brown part should be on top. And then what should happen to those blue portions? They should now be on the bottom because the blue is going to fuse with the brown, or the caudal portion is going to fuse with the rostral portion. What is this called? Resegmentation. Why is it important? To allow for the intervertebral spaces for the spinal nerves to run through. Boom diggity. All right, and again, that'll be allowing for these spinal nerves, once the actual nervous system develops, for that to run through. Isn't that cool? And then what happens? So now, imagine, here's your somites. They went through the resegmentation process where caudal fused with the rostral. Okay? And in this situation, what happens? You get the resegmentation. Then this sclerotome, that component of the somite, is going to start doing what? Forming bone tissue, right? Via the process of endochondral ossification around the neural tube and the notochord. How does that process work? Ba-boom! Look at this. Isn't this insane? So then once you have that here, all of this actual sclerotome is going to start surrounding the neural tube, which will become the spinal cord in this case. All right, so now we went through the resegmentation process. Now, again, what are these actual sclerotomes going to start doing? They're going to start moving around this actual neural tube and the notochord. And when they start doing that, they're going to start forming the vertebral arches, which are going to kind of wrap around this actual neural tube. So now you're going to have a vertebral arch right here, and then you're going to have a vertebral arch over here. They're going to fuse. And when they do that, you're going to get this little point here at the end called your spinous process, right? And then what else is going to happen besides forming that? They'll continue to form in the anterior portion here. So when they form around the anterior portion, what else are you going to get? You're going to get the vertebral bodies and some intervertebral disc there as well. Then coming off of this portion, you're going to have what's called your transverse processes, right? But then bone will continue to grow from that transverse process and a little bit of the body. What is this going to be called? These are going to become your ribs. So from this sclerotome, which is formed from the paraxial mesoderm, which undergoes this chunking process called forming somites, resegments. Then from that resegmentation, the sclerotome of those somites surround this portion and make the bone or the vertebrae and the ribs coming off of it. Isn't that cool? So that's that part there. Now, let's make the sternum and connect the vertebrae and the ribs to it. Where does that come from? All right, so we formed our vertebral column, right, from the sclerotome component of the somites. We went through the process of resegmentation, and then we talked about how that completely sclerotome portion of the somite made the vertebral column ribs. Now, let's go ahead and make the sternum. This part's a little interesting. So we have to kind of take a quick look back over here. So remember we had this lateral plate mesoderm? Remember we had two components of it. One here, this component here, I'm going to kind of asterisk that part. That's the splanchnic layer of the lateral plate mesoderm. And then this portion here was the somatic layer of the lateral plate mesoderm. Where do you see this kind of like lateral plate mesoderm kind of coming? You see it kind of like right here. That is the anterior portion of the actual what? That's the anterior portion of the embryo. So if this is the anterior portion of the embryo, guess what that lateral plate mesoderm is going to do? This lateral plate mesoderm is actually going to do what? The mesodermal cells will then convert into cartilage. So here we're going to have our cartilage kind of like model here that we're going to go from. Now, when we had that differentiation from the mesodermal cells to the cartilage model, that was one step. Now, that cartilage is then going to start converting into bone. What does that process called? Mesodermal cells to the cartilage to the bone. This is called endochondral ossification. You're getting the point, right? That endochondral makes up most of the actual bone formation. Now, this process where you make the bone, we're going to have this cartilage model. It's going to lead to bone that's going to fuse and form three components of the sternum. What are the three components of this sternum? This whole thing here is your sternum, but it's made up of 3 points. This part here is called the manubrium. This is called the manubrium. It's fused to the body via what's called the sternal angle or the angle of Louis. And then you have this xyphosternal joint here, and that is going to be connecting the body to the xiphoid process. So then you have your xiphoid process. This is all going to make up your sternum, which is going to be coming from the lateral plate mesoderm to the cartilage model to this bone. Then, if we take the sternum, this is in a three-dimensional space, right? This is formed from the lateral plate mesoderm. Imagine from the paraxial mesoderm, which part? Sclerotome, you made the vertebrae, you made the ribs. The ribs are starting to come around, grow. Connect to what? The sternum. Ba-boom, babushka, right? So now we had the ribs formed from that actual paraxial mesoderm, the vertebral body, and all the intervertebral discs connecting to the sternum. We just made our what? Trunk. Let's move on now to the limbs. All right, so we formed so far the skull, we formed the trunk. Let's finish this up strong, ninja nerds. Let's go into the limbs. The limbs are going to develop from 2 components of the mesoderm. So far what we've seen is we've seen paraxial mesoderm involved a lot. We've seen some neural crest cell, and we saw a teensy bit of the lateral plate mesoderm. Well, the lateral plate mesoderm is coming back again. So what happens? Well, in order for us to start forming limbs, what happens is we have to first know, where do I want to put the limbs? That's the first thing that I need to do. Where do the limbs need to go? Okay, so I need to know the position of my upper limbs and the position of my lower limbs to grow from the embryo. All right, we're calling them limb buds. So in other words, I need particular genes to get activated in order for me to find the position to start initiating the formation of limb buds. What are these genes? It's very important that you know these genes. These genes that'll lead to kind of the starting of the position. So the position of the limb buds, kind of getting this whole process initiated, is going to be your Hox genes. So you have different types of Hox genes that are going to be activated. Okay? Other genes that will be activated in this process are going to be what's called TBX genes. And the TBX genes will kind of determine a little bit, particularly the formation of where the upper limb bud's going to come and the lower limb bud is going to come. So the position will be kind of started and initiated by the Hox genes, but the TBX4 and TBX5 genes are really what's going to determine the position of the limbs. Which one's going to be upper, which one's going to be lower. When these genes are activated, they then do something very interesting, which is they help to lead to the actual development or differentiation into particular types of, they lead to the production of particular growth factors that will be released in high concentrations. And this is called fibroblast growth factor 10. So these genes will be activated for position. This will be kind of activated for knowing which one's upper, which one's lower. And this one will be secreted for a particular reason, which is allowing for the actual continued growth of the limb buds. And we'll explain how. So here we're going to have the upper limb buds, and here we're going to have the lower limb buds. We're going to abbreviate them for simplicity's sake. So here's going to be our upper limb bud, and here's going to be the lower limb buds. Now, these are going to be the ectoderm. So you obviously know this is our ectoderm, then we have our mesoderm, and then we have our endoderm, right? So the ectoderm is going to kind of grow off here, these little limb buds that are going to come off. Now, which part of the mesoderm do you see has quick, easy access into the limb bud? The lateral plate mesoderm. Which component of the lateral plate mesoderm? You guys better know it. This is which one? Splanchnic. This one is the somatic layer. So what is going to happen is this somatic layer of the actual lateral plate mesoderm will start moving into these limb buds. And when they start moving into the limb bud, something interesting starts happening. Let's talk about that. Okay. That fibroblast growth factor 10, what it does is it stimulates this little area here at the tip of the limb buds. We're just zooming in. I'll look at this limb bud here. Okay, we're just going to zoom in on this one. And we're going to only focus in on this limb bud. When we look at this limb bud, there's this little thickened area on the tip of that actual limb bud. And this is the ectoderm. What is this thickened area here called? This is called the apical ectodermal ridge. Okay, commonly abbreviated the AER. What happens is the fibroblast growth factor type 10 is going to stimulate this apical ectodermal ridge. What the apical ectodermal ridge will start doing is it starts actually secreting other growth factors. And when it starts secreting these growth factors, it starts stimulating this lateral plate mesoderm that invaded into the limb bud to start proliferating. What are these growth factors? This one secretes two types of growth factors. One is called fibroblast growth factor type 4 and fibroblast growth factor type 8. And these are going to start initiating tons and tons of proliferation of the lateral plate mesoderm. Now, when it does that, the portion of the lateral plate mesoderm that is closest to the apical ectodermal ridge will get hit with so much of these growth factors that all they do is proliferate. They don't have time to differentiate. That's very important. So this area, let's kind of imagine that I kind of do like this. Let's kind of say just this chunk here, okay? This chunk here of the lateral plate mesoderm that is closest to the apical ectodermal ridge, this is going to be proliferating a ton. So we call this the progress zone or the proliferating zone, sometimes abbreviated as PZ. Now, as these cells, these mesodermal cells start proliferating and proliferating and proliferating, this allows for what happening? The limb bud to continue to grow from proximal to distal. So what is allowing for proximal to distal growth? Apical ectodermal ridge and these growth factors interacting with the progress zone. I can't stress that enough. That is for proximal to distal growth. Now, as the limb bud continues to grow from these growth factors, remember what I told you. Pretend here for a second. I'm going to kind of erase this for a second. This was just for a second, that was that lateral plate mesoderm, right? And then this portion here was our progress zone. This portion here was our apical ectodermal ridge, releasing those growth factors. We said the mesoderm closest to that proliferates. But what about the mesoderm that's far away from the apical ectodermal ridge? What happens to that? Well, growth factors are really saturated in this area, but they're not really saturated much in this area. So they don't have time to proliferate here, whereas they're proliferating a ton here. So if they don't proliferate, guess what they do? They differentiate. And if these mesodermal cells differentiate, what do you think they differentiate to? Cartilage. Then from cartilage to bone. What is that called? Endochondral ossification. So what are we going to make? Bone. And this bone here that we would have just as a kind of an example here would be our humerus, right? If we're looking at the upper limb bud. So that is this process. Now, let's come down and talk about what happens next to the limb bud. All right, cool. So far what we've done is we've gone through this process of where we've done and made bone from the actual differentiating mesodermal cells and not the proliferating ones. Now, another thing that starts to happen, okay, again, let's kind of fast forward a little bit. We had the humerus. Here was our progress zone. What happens? The limb bud continues to keep extending from what direction? Proximal to distal. So now here's our new progress zone as this limb bud continued to keep growing. This was one part, which was the humerus, right? You know what they call this embryologically? They call this the stylopod, because technically the limbs are developed in 3 components. Now, this progress zone continued to keep growing. So technically, if you wanted to think about this right here would have been that kind of lateral plate mesoderm that wasn't having a lot of proliferation. So it did what? Differentiated. What did it differentiate to? Cartilage. Then it differentiated from cartilage into bone. What is that called? Endochondral ossification. This portion here is called the zeugopod, even though technically we just call it the, the forearm, right? Now, the next thing that happens is here's your progress zone. Here's the apical ectodermal ridge. It's still thickened here. We'll thicken it even more here for you. This is still thickened, but what happens is some of the tissue in the apical ectodermal ridge starts actually undergoing apoptosis. So there's some little bit of apoptosis here in between the apical ectodermal ridge portions. And this is due to high levels of bone morphogenic protein and retinoic acid. These levels, the change in these levels lead to apoptosis of some of the cells in this apical ectodermal ridge. And it kind of starts doing what? Forming what looks like fingers, right? Now, there's still apical ectodermal ridge here. So it's still releasing what? Fibroblast growth factors, type 4 and 8. So that's still telling the actual progress zone to proliferate. So where does that progress zone start proliferating into? Into these like little digits, what looks like it, right? Now, as you again come down here to the next part, here was the progress zone and kind of like the wrist and finger region. What do we say? As you get farther away from the apical ectodermal ridge, what starts happening to those mesodermal cells? They differentiate into chondrocytes, then from chondrocytes into bone tissue. And now we're gonna have kind of the carpal bones. Then from there, what else is gonna happen? Then this actual progress zone infiltrates into these like little digital components. And that is going to start making your metacarpals and your phalanges that are going to extend into this. Now, how this happens though, how do you just prevent the fingers from continuing to grow? Well, guess what happens to the apical ectodermal ridge? The apical ectodermal ridge in this next step here starts dying off. It starts dying off. And as the apical ectodermal ridge dies off, what happens to the fibroblast growth factors? They drop. What happens to the progress zones? They don't keep proliferating. If they're not proliferating, what are they doing? Differentiating. And eventually, this converts into bone and no more growth will be occurring here from proximal to distal origin. Now, that covers the proximal to distal growth of the limb bud. Big things you need to remember, apical ectodermal ridge is driving the progress zone. How is it doing it? By these fibroblast growth factors. Now, apical ectodermal ridge starts breaking down, you get the digits, progress zone infiltrates it, you get bones there. This last portion here, right, we said stylopod, zeugopod. Technically this last portion here is called the autopod. And again, we just like to call it the wrist and the fingers, right? But that's kind of the embryological term for this kind of development. Now, we talked about proximal to distal. Now what I want us to do is hit anterior to posterior development, or a simple way of saying it, how does our thumb look this way and look on this side, and our pinky this way and on this side? All right, so we formed pretty much the limb bud, right? We went through that entire process from proximal to distal. So we know how to actually extend the limb bud out. Now, how do I make my hand look the way it does, particularly from the thumb to the pinky? How do I get this kind of configuration? Very interesting. So I want you to imagine, obviously this isn't the most perfect thing, but I want you to imagine all these fingers in this diagram are exactly the same. They look all alike. We don't know which one's the thumb, which one's the pinky, right? Well, we have to start developing that process. So on the actual kind of like dorsal portion here, during embryological development, this is kind of the posterior portion. There's a little condensation of tissue here called the zone of polarizing activity. We call this the ZPA, right? But we'll write it out here, the zone of polarizing activity. This is what drives the anterior to posterior development of the hand. In other words, the thumb on one side, pinky on the other side. How does it do this? Well, it starts releasing particular types of molecules that kind of create a gradient throughout the actual hand region, right? Particular types of molecules that create a gradient, and those proteins or growth factors that are actually there, depending upon that gradient, creates the growth of this distinct differences in our actual fingers on the hand. All right, so what are the genes for the dorsal aspect of the hand? That's going to be this Wnt7a genes. And these Wnt7as will actually induce the activation of what's called LMX1. And the LMX1, guess what it's going to do? It's going to lead to stimulating the dorsal hand growth. in a specific type of way. What way can we distinguish the dorsal aspect of the hand from the ventral? The nails, right? So it's going to kind of lead to the particular kind of growth of the nails and other aspects of how the dorsal aspect of the hand is grown. The ventral, right? The ventral is by this process of engrailed 1 genes. When these are activated, they are going to stimulate the ventral growth of the hand. And what are one way that we can kind of distinguish that? The ventral hand growth is going to be via what process? This is going to be from the actual fingerprints. Now, what is really important that I need you guys to know here is that the dorsal and ventral aspect of the hand, this is primarily mediated by what tissue? What do you notice here? Did you notice any mesoderm involved in this? No. This dorsal and ventral aspect of the hand is carried out by the dorsal and ventral ectoderm. I need you to remember that. So this is actually carried out by the ventral ectoderm and the dorsal ectoderm, not the mesoderm. All right, engineers, so let's finish up talking about intramembranous ossification and endochondral. We know what bones are developed from which ones, right? It's pretty straightforward. Endochondral is pretty much all the bones in the body. The only ones for intramembranous are which ones? Do you guys remember? The top of the skull, and what else? The facial bones and the clavicles. That's it. Everything else is endochondral. The last thing I want you guys to know, though, is how, because this is important to think about. Yeah, we know the process, but how do these cells turn into these cells, turn into these cells? It's based upon the genes that they're expressing. So whenever these particularly mesenchymal cells, right? So these mesenchymal cells, whether this be from the ectoderm, whether this be from mesoderm, the paraxial, the paraxial mesoderm, the neurocrest cells, whatever. These mesenchymal cells have to activate particular genes to turn into bone cells. What are these genes that they've seen most commonly involved in this? The genes that you most likely see that are involved in this process are what's called the CBFA1 genes and the RUNX2 genes. Whenever these are activated, they lead to particular proteins that give the phenotypic development of what types of cells? Osteoblasts. Okay, and obviously osteoblasts have other phenotypic variants that allow for them not only to look different, but to function differently, produce different things like osteoid and different types of collagen, so on and so forth. Same concept here. So we obviously know that the CBFA1 genes are important for the development of, and RUNX2 genes are important for the development of the bone cells. But what about the conversion of mesenchymal cells to chondrocytes? So going from mesenchymal to chondrocytes. Because we already talked about the osteoblasts. We already know what genes are primarily involved in this process. What about the chondrocytes? There's particular genes here, and the main gene that's involved in this is called the SOX9 gene. And the SOX9 gene, whenever this is activated, leads to obviously the complete phenotypic development of chondrocytes with the ability to produce different types of proteins and molecules that distinguish them from other cells. This is going to pretty much give you everything that you need to know for the development of the skeletal system. All right, Ninja Nerds, so in this video today, we talk about the development of the skeletal system. I hope it made sense and I hope you guys enjoyed it. All right, Ninja Nerds, as always, until next time.